Thank you so much, and thank you for sacrificing a beautiful spring morning. I am from Sabancı University, so this is our campus. I suggested that we do these satellites there, but Can Alkan, our chair, said that it's not counted as Istanbul because we are at the edge of the Istanbul. He has a point. This is Beşiktaş is a very strategically nice place to see around Istanbul. I hope you will have time to do that. So today, I'm going to be talking about some recent work that we have been doing in our lab. So there's going to be three parts. The first part, they are going to be different cancer-related problems, but the common theme is going to be kernel methods. The first part is going to be on patient subgroup identification, and the second and the third parts will be related to detecting regulatory interactions between non-coding RNAs, coding and non-coding RNAs. So basic machine learning 101, we have the kernel functions that measure these are functions, special functions that measure the similarity of two objects using an inner product. I'm not going to get into the technical details of it, but the nice thing about them, the most familiar ones, if you have ever run SVM, people call SVMs now ancient tools, which makes me a little sad. But if you have ever run a kernel SVM, you would know we have the linear kernel, Gaussian kernel, polynomial kernel. But these functions measure the similarity of any two objects. These objects can be documents, they can be cats, they can be graphs, they can be trees, they can be anything. It doesn't necessarily have to be just feature vectors. And it's possible to, in the literature, we have seen a lot of versions of this. It's possible to kernelize classical methods. For example, we have kernelized classifiers, we have kernel PCA, kernel k-means, or kernel statistical tests. So in this talk, we'll be using some kernel clustering methods and kernel interaction tests. So why do we like them? Kernels has this property that they implicitly map the data into a higher dimensional space and compute the inner product in that higher dimensional space. We don't have to do this explicitly. So the kernel function will return us a value as if we had projected our data into a higher dimensional space and get the dot product. So here in this picture, the classical example of getting, this is the XOR problem. We map it it's originally in 2 dimensions. We map it to a 3rd dimension where we have the multiplication of the original 2 dimensions. And suddenly the data become linearly separable. This is an example of how we use this in classification. In PCA as well, if we have nonlinear data, if you were to apply just simple linear PCA, you might not be able to get that separation well, but if you kernelize the method, then you will be able to do that because it maps it to this higher dimensional space and so on. The kernel matrix, like in the original methods, we use this, the representation in the original feature space, but when in the kernelized methods, we use the kernel matrix, which is for every pair of objects that we have, we apply the kernel function and get a value and put them into a matrix, right? So, and the kernelizing usually involves replacing the original dot products in the original space with the kernel function. So, for example, when we are doing this with the kernel k-means, whenever we see a dot product, we replace this with the appropriate kernel function value, and then suddenly we don't now rely on the original feature space, but just the kernel function. So this is going to be kind of the common theme of all these 3 projects. Let me start with the first part. This is a published work, but we have still ongoing things to improve it further. This was mainly done by Yasin and Ali Burak Ünal as part of their thesis. Beyza later focused on improving some parts of it, and Ali and Furkan helped along the way on some technical parts of the project. So for this audience, maybe these slides are too basic. So we know we have this traditional medicine where we try to find cure for the cancer for the average patient, but we also aim for the precision medicine where we tailor the drugs or the therapies based on the patient's genomic profile or the medical history and so on. But there's something in between called the stratified medicine. That is, we identify the groups of subgroups of the cancer patients that are homogeneous in terms of their molecular profiles and try to target them. I mean, this is good to devise better treatment strategies, to do better diagnosis for the patients, and also to get insight into molecular mechanisms of the subtypes, because of this heterogeneity, it is difficult to pinpoint what are the molecular underpinnings of the cancer. But when you have better subtypes, it's easier to work with. And in this work, we use two ideas, and neither of these ideas are novel. People have been doing this, but we put them in a framework using kernels. So the first idea is using multi-view clustering. So we have all these omics technology coming up, which are measuring different parts of the tumors, profiling the tumors in different aspects, their mutations, their expression profiles, and so on. And in clustering, this is, whenever we have this type of data, in clustering, this is studied under the name multi-view clustering. So like shown in this picture, you might have different projections of the truth. And if you combine them, you can get a better understanding of what should be the truth, right? So this idea has been applied a lot in the cancer field, as you might have known, under the name of multi-omics integration or multi-view clustering and so on. And we have many methods, which is reviewed nicely in this paper by Maria Ziklik. So we use one approach under this, which is multi-view kernel learning, and this relies on, so we have different, we are going to get different views of the data. This is the general multi-view kernel framework, studied also outside of the biology domain, where people work with images, text, and stuff. So you would have different views of your data, and you would compute kernels on these different data types, and then combine them through a kernel learning procedure to get a unified kernel. And this usually involves like finding the weights, if it's a linear combination, finding the weights of these kernels to arrive at the final kernel. So in our case, so we will have data of the patients and their features. These features are going to be their mutations or the expression profiles, and now we will arrive this patient by patient kernel matrix. So this is one idea. It's old. Like let's combine different types of data, the mutations or the expressions, right? And the second idea is like these tumors are quite different from each other. For example, if you were to look at the mutational profiles of the cancer tumors, we often see that very few genes are frequently mutated across many patients, but there are a lot of genes that are mutated only in few patients, right? So one way to handle this, but we also know that molecular interactions orchestrate biological processes, right? And one idea that's been used was like to look at these changes in the context of the biological interactions. They could be protein-protein interactions or the pathways, right? So that's the second idea. So, and the reason is that, like, it might be the case that, let's say we have these schematic representations of pathways where everything is just shown as nodes, and the edges nodes are the genes, and the edges exist if they interact, and a color indicates that there has been an alteration in that patient tumor. So it could be the case that the identical genes are not altered in a given two patients. For example, when we look at patient 1 and patient 2, there is no identical mutations. Similarly, like 1 and N is quite different. When you look at patient 2 and patient N, there is, 5 is not mutated, but 4 is a neighbor, 8 is a neighbor that's mutated, and 9 is a neighbor, and so on. So if we were to just base our similarity, assessment of the similarity between the patients on the identity of the changes, that is like look for identical genes, number of identical genes that has been altered, this would have been only 10, right? Neither the similarity due to the 5 and 4 or 9 and 8 would have been counted. That's the idea, to put them in the context of the molecular interaction and then look at the similarity. But how are we going to do that? So let's get the multiviews from omics data and multiviews from different pathways. And as I said, these have been studied a lot there's lots of papers that do either of these. Now what we did was, so we developed this framework, and I'm going to walk you through how we do it in this multi-view kernel framework. So the first, we are going to generate multiple views of the patients per omic data and pathway, and per pathway. So per pair of pathways and omic data, I would say. So that is for every patient. So let's take patient 1. For pathway 1, we downloaded cancer pathways, turned them into small graphs, undirected nodes. That's simplifying biology. So the undirected graphs, and only nodes represent. And these pathways are much smaller, of course, than the original pathways. They are like hundreds of nodes, but to fit in the slide, we have these small pathways. What we are going to do is we are going to map these alterations on these pathways. So for a patient, for all the pathways, I will map the mutations of this patient on the pathway. I will do that for the protein expressions as well. Like if this expression level has altered in this patient, it's going to get a colored node here. And for gene expression as well. So among the different data types that's been produced, we worked only with the three of them, because when you look for a patient where all the measurements are there, the data set becomes smaller and smaller. This was keeping only to 3 lead to us have a sizable cohort. So at the end of the day, for each patient, for each omic type, and for each pathway, we have one graph, and this graph is node labeled. So this is the structure of the graph for a specific pathway is the same, right? The pathway doesn't change. But the mutations or the alterations are patient-specific. So now, for a single patient, we have a set of graphs, node-label graphs. And for the other patient, I have another set of node-label graphs. So how am I going to compare these graphs and get a similarity out of it? That's the question. And this has been also studied. Like people have looked at, under the name of graph kernels, people have looked at the question of like how you can compare 2 graphs and compute a graph kernel that will return you a value. If these graphs are similar, it will be high. If not, it's going to be small. And there is a lot of them. And there is this encyclopedic paper from a review from Karsten Bogwart group, where they have nicely categorized all different types of graph kernels that's been developed to date. So why to bother? I mean, we can just pick one and then live our life, right? People have studied this, and these are really nice papers with good theoretical results and so on. But these are propagation, Weissenstein, Weisfelder, Lehmann graph over. These are like top 3 good kernels. So the problem is that they are solving a much more difficult task. Like they are trying to assess. You give 2 graphs. These 2 graphs doesn't necessarily have to have identical topology. The way they work is like to figure out how the topology is different or similar between these 2 graphs. In our case, the pathway is the same, only the node label distribution is changing. So if you were to apply an existing graph kernel, and this is a heat map showing the kernel values of the patients, so this is rows are patients and the columns are patients, and the color, a dark color indicates that they are highly similar. So if you were to, for example, take a propagation graph, apply it to these patient pairs, this is on a specific pathway with the expression data, you would find out that everyone is similar to everyone, because these graph kernels are solving this difficult problem of how similar the graph structures are, but the graph structures are going to be mostly similar because the pathways are similar. So we need something, a slightly different thing than the existing ones. So we want to compare the node label distribution, but taking into account the topology of the graph, so the neighborhood information of these changes and so on, right? So So the bottom line, in the existing graph kernels, when we tried them, like we would be happy if one of them would have worked, we would we would go with it. They all evaluate them very similarly. So the question is, same graph structure, different node label distribution, how would you assess them? So we came up with the strategy which relies also on previous ideas of like propagation label or propagation kernel or diffusion kernel. So the idea is, first we propagate the alterations on the graph using random walk. So if there is a change in a gene, then the neighbors also receive some amount of change. So this step brings the context into play, right? When you diffuse your labels, you now take into account the changes in your neighborhood. And then we check how, so in most of the graph kernels, the way to assess the similarity is to look for substructures, either graphlets, walks, or paths on the graphs and compare those. We do a similar thing. We find all the shortest paths among all pairs in the graph, and then we compare the node label distribution. So this SP for patient 1 and SP for patient 2 these are two vectors that is telling me on a given shortest path what is the node label distribution and what is the node label distribution on the same shortest path, but for the second patient. So if these node label distributions are similar, it will contribute that these two patients are similar with respect to the changes in this given specific pathway. So, and when we do that, we would end up with having lots of kernels. So one for each pathway and the omic type, right? So we have hundreds of pathways. Here we use 165 pathways, so multiply that with 3. Actually multiply with 5 because we express the upregulation and downregulation differently. I'm not going to get into the detail, but it worked better than just taking the expression as is, the changes as is. So we have like hundreds of pathways, and now we need to integrate them to get the clustering, right? So multi-view kernel clustering is something that's been studied a lot in machine learning. And this is, at the end of the day, this is how we are going to evaluate the models. This is not perfect, but we look at you find your clusters, you have your patients, and you get the survival distributions of these patients, and then compare how different these survival distributions. If there's significant difference among the cluster survival distributions, you say that, okay, maybe this is clinically interesting, right? So, and to do that, we use the Kaplan-Meier curves and log-rank test. And the p-value, the smaller the p-value, the indicative, it's indicative that these curves are more separate than each other. So now the next step was like which multi-view kernel clustering method to use. So we tried different things. So this is, so here you can see the references. This one might be familiar, similarity network fusion, published in Nature Methods, and applied to cancer, so you might have seen this work. This is similarity network fusion, where we replace the spectral clustering with the k-means. This is localized multiple kernel k-means by Mehmet Gönen, and this is the simple average kernel k-means. You just compute the kernel k-means for kernels for each of these graphs, and then take the average and input to the multi-view kernel clustering method. And we have the multiple kernel k-means with matrix. So the bars here, the higher the bar, the lower the p-value, that we are doing a better job. So this is applied on kidney renal cancer, Kirk. And here we find out that this one, multiple kernel k-means with matrix index regularization, does a good job compared to the other ones. And the nice thing about this method is, what it does is it tries to assign weights to the kernels that it's combining. And these weights are indicative of like which kernel has been useful in my clustering. And that's going to help us to interpret which pathway or which kind of data type has been useful in the clustering. So The other thing that we evaluated was like, if we were to use other graph kernels, what would we have get? And we call this smoothed shortest path kernel, and it does better than the existing ones. Here, RBF kernel, radial basis function, does surprisingly well. We just compute the take the alteration vectors and then compute the radial basis function. But it takes all the genes. When you map the genes to the pathway, some of them will be left out, right? Because not all proteins are studied well. So this takes all the genes. These others don't, because they work on the pathways. So then we did we bootstrap our inputs, like instead of the original one, let's generate different versions of the input and run the methods and to compare them a little bit better. So these box plots are showing the p-values that we have, log p minus log p values that we would get when we run each of these methods. So the PAMOGK is the one that's up here. SNF, for example, this Nature Maths paper is here, and so on. So these are all different methods from this nice benchmark paper from Nucleic Acids Rappaport and Shamir. They have nicely summarized different methods, and we used implementations also that they pointed to. The next thing, so when we apply to this kinder cancer, that's finally the clusters that we get. We also checked whether these clusters could be happening for other reasons, right? Maybe because we are figuring out clusters of different genders or age and stuff like that. So we checked is the clusters we tested for if there's a relationship of these other clinical parameters. It is not the age, it's not the gender, but these other important clinical parameters are also associated with the clusters, which is not a bad thing. We also analyze like the these clusters proportions of in these other different clinical parameters. How much time do I have?
10 minutes probably.
Oh, okay. Part 2, okay, I will be running. It's okay. Yeah. Okay. Okay. Yeah. So, as I said, we can interpret the pathway importances with this method, and this is due to the multiview kernel method that we are using. They provide us the weights, and we can look at the kernels and sum them over the omic type and get how important was each pathway for the specific clustering, or we can do the same, sum over the pathways and get the importance of each of the omic data types. So it turns out here, like the P53 effectors, of course, turned out to be important. And but this ERB1 bounce, this one, especially HIP1 alpha transcription factor network, this is more related to kidney, and we were able to recover this one. Okay, so we also, Beizal, worked on trying to improve this method, or enrich it with other networks. Like, what if instead of a pathway we were to use the tissue-specific protein-protein interaction networks? The kernel that we compute is not going to work with the protein-protein interaction networks because you need to find all shortest paths. So we devised there something slightly different where we find communities first and then compare communities inside it. We also enrich these pathways with high C-based gene-gene interactions and so on. And like preliminary results, we are seeing that sometimes, like when you have these protein-protein interactions augmented with high C-based gene-gene interactions for other cancers, they are doing better than the one that we have done. So you can reach this paper and the code on GitHub. I'll be quick in the part 2 and part 3. We will give you the kind of the basic ideas of what problem we are doing, solving, but we can talk about them maybe in the breaks. So the second and the third part deals with the noncoding RNAs, and so the first part is sponge interactions. This was brought to our attention a colleague, Özgür Şahin, who is an expert in breast cancer, experimental biologist, said that there is these sponge interactions. Will you be able to discover them? So in 2011, Kasena et al. Published this work. They showed that a long non-coding RNA controls the muscle differentiation by function as a competitor to as a competitor. And how does it do this? It sponges these microRNAs. So microRNAs binds to mRNA. They repress the expression of the mRNAs, right? So this lncRNA has binding regions to the microRNAs. So normally, these in the existence of these microRNAs will down-regulate these genes. But when there is the long non-coding RNA, these microRNAs will bind to them, and then suddenly there is no more enough microRNA to down-regulate these ones. So they kind of indirectly, and this picture might be a little bit better. So microRNA binds to the mRNA, but if you have the lncRNA in there, it might sponge away the microRNAs, and then you don't have down-regulation for these mRNAs anymore. So can we discover these? People have looked at this using looking at the correlation and partial correlation coefficients. We formulated this problem using kernel-based conditional independence tests. That is like, we test for so the input to this is, we have matched expression for the patients. We have microRNA expressions measured for all the microRNAs, and then we have the mRNA expressions for the same patient. Then and the lncRNAs as well. We look for if the lncRNA and the mRNA expressions, is there dependency? And for this, we use the kernel-based conditional independence test. Is there a dependency between these two expression levels? And then we test for conditional independence between lncRNA and mRNA. So if this relationship is related to the microRNA, if we condition the test on the microRNA expression level, they should become independent, right? So we look for pairs like this. The whole pipeline is a little bit involved. Like we also take into account the target interactions that is deposited in the databases and so on. But for example, this is one example that we found out. So the method doesn't take into account any genomic information, like these microRNAs on this chromosome and this lncRNAs here, mRNAs. We don't input this information, but for some of the things that we discovered, and this was one of the interesting ones because they are relevant to cancer, we applied this to breast cancer subtypes because of Özgür's interest. Like we figured out this hot-air lncRNA and the HOX-C genes and this miR119.6A. They are in frequent sponge interactions, like there are multiple sponge interactions that we discovered. And when you look at their genomic coordinates, they are actually also in the vicinity of each other. So that kind of provides some indirect evidence that they could be actually working together to regulate some biological processes. We can also do prognostic analysis with these sponges, like are these the expression level of the sponge interaction different across, when these sponge interaction expressions are different in two groups, will their survival distributions be different? Like we did this analysis as follows, like you take a sponge interaction, there is an mRNA, microRNA, and a lncRNA. If you were to group the patients based on single molecule, so is this mRNA upregulated or downregulated, and you have two groups, right? And look at the survival distribution, there is no difference. You look at the microRNA expression, group them, there is no difference. You look at the lncRNA, there is no difference. Sorry, I went on the wrong order. But if we do that based on the sponge interaction, so we expect that when lncRNA is up, microRNA should be down, and mRNA should be up in this interaction. If we do this grouping based on that, we see a significant difference. So we have this bunch of list of sponges that could be interesting, waiting for experimental biologists to work with, but we didn't get yet those. So Özgür was the one who gave the idea, and Gülden did this work as part of her thesis. And should I be done in 2 minutes? Okay. Okay. So, and thank you. So the last part is also related to the non-coding RNAs. So this is, again, going to be using some interaction tests. And we are also actively working on, this time experimentally, not me, but my collaborator, Özden Konu, and our students are working to try to validate some of these things. And Gülden and Öykü and Melike also worked on the computational parts. So here, so it's been reported that microRNAs replace their target mRNAs only modestly by themselves. But there is a lot of cooperativity going on among them, and it could be in different ways. They can be targeting different constituents of a pathway and so on. But one type is the different microRNAs can bind to the single mRNA to repress more. So synergistically, so in a stronger fashion to repress the target. And we thought that this problem, so here what we have is you have by themselves, they are not repressing much, and this is reported actually in literature, and there are other examples of this kind of behaviors. So individually microRNAs are not strongly doing it, but together they are inducing a stronger effect. This actually related to a problem called restructure discovery in machine learning. That is like you have, let's say we have 3 variables, x, y, and z. X and z are independent or weakly dependent. So when you look at this graph, for example, here, that's what you see. So imagine x and y are drawn randomly from a normal distribution. And z is drawn from this distribution. That is the sign of x multiply y. So it's definitely z is dependent on the x, y. But when you look at individually, neither y or z, x is strongly dependent to z, right? X and y also are not. But x and y together, they are actually not independent from z, because as you see, this is how it's generated. So this example is from this paper, which is also the author of the kernel interaction test. So this is similar to like you have your tea. I hope you will get to enjoy some tea in Istanbul too. You should, with simit. So you have your tea, you put some sugar on. If you ignore the diffusion and the hotness of the tea, by itself, it won't make the tea like immediately very hot, right? So we have 3 variables: putting sugar, stirring the tea, and sweetness of the tea. By themselves, neither putting sugar alone or stirring alone will make the tea very sweet, right? But if you put the sugar and then stir it, it will make it very sweet. So this is similar to that. So we have like weak individual dependence, but together they are dependent to this variable. So this is actually studied way in 1969. There is this Lancaster interaction test, and Sejdinovich and Greton, they work to kernelize these tests. So we thought, I happen to know Arthur from CMU, and I thought, okay, this problem is like very similar to what we are trying to solve, so we imported their solution to here. So what we are doing is, in our case, these X and Y are microRNAs, and mRNA is the expression. So here we developed a pipeline. So the pipeline will take first, we compiled lots of data from microRNA databases on the interactions, microRNA, mRNA interactions, who interacts with whom, so that we can sort out our list. Because if you take triple combinations of all mRNAs and microRNAs, that's a huge number, like hundreds of thousands of things, right? So, and then we have the cancer patient expression data, which we are going to use to test for these dependencies. And we put them into our algorithm. So the algorithm will check whether these microRNAs are actually targeting the same mRNA, make a short list, and then we check for expression levels. Like when they are both up and down, there should be significant difference in the mRNA level. And then finally, we would input this to the kernel first pairwise interaction test to check mRNA, microRNA, mRNA dependencies, and then 2 3-variable interaction tests. And from this, we get a list of things that could be possible triplets that are in interaction. Doing this was extremely time-consuming because you have hundreds of thousands of tests. And we have our HPC gurus at Sabancı University. They helped us to parallelize these things. So we have like now 5,000-fold increase speed up in the implementations of the original implementation of this kernel test. So we were able to run this on 31 cancers. So soon, I'm not going to get into the details of these, but we now run this on all these cancers and have the lists, and the lists will be coming up on this server soon. This work is not published yet. It's almost ready, the web server, but you can still go and play. And if you have any feedback, please let me know. But you'll be able to reach, for each cancer type, what are the identified synergistic interactions. And also, if you select multiple cancer types, you will be able to see what are common between these cancers. So I'll stop here, and thank you so much for your time. I thank to my my students and collaborators, and also our funders. I'll also make a little advertisement before stopping. Tomorrow, Halil, who's sitting there, is going to have a talk on personalized drug synergy prediction. This is some deep learning method, so fancy. So you can go and listen to that one as well. Thank you so much.
Thank you. Great talk.
Thank you.
Great way to start the conference. We have time for a couple of questions also because there will be a program change in the next session, right?
Okay.
Yes.
So,
so just before the questions, did the speakers were able to upload the slides? No.
so who do we need?
You can go to the questions.
Right. So we have time for a few questions, and I'll start.
Or
the chat.
yeah.
So I kind of have a memory from like 10 years ago, which people were kind of giving up on
Kernel
learning
methods.
methods because they were saying any combination of the kernels will give you roughly the same performance. This was a very different supervised learning topics. But I was wondering how stable you find your kernel weights in your experiments, because that's kind of a crucial.
The weights, actually we haven't checked that. That's a very good question. So we have checked how stable are results in terms of the p-values, but we didn't check how stable are the recovered, or did we? No, yeah, we didn't. Yeah, at one point I think we talked about this, but then rushing through the paper we didn't. But that's a good idea because, but I mean, we have run these experiments a lot, so with this method, like in most of the multi-view kernel methods, the challenge is like, the original methods are all work with like 3 types of kernels. Like you have image, you have text, and so on. Here you have hundreds of things, and they break down when you put hundreds of kernels inside them. And only like this one, the one that we used were good. And then the weights also, I think, I mean, this is not quantitative, but we're not unstable, I think. Yeah.
So you think Trevor had a question?
Yes. Lovely talk. In the first part, have a reliance on knowing the structure of the graphs. Like how do you handle uncertainty in the graph structure, or the fact that the graphs probably change when you get mutation?
Yeah, that's a very good question. I mean, right now we don't handle that, but once we have these predictions or maybe actual experimental data on personalized graphs, I think that would be something to deal with, right? But we could maybe do some experiments, simulated experiments, like if we were to change certain edges, what would have happened? Or what you had in mind, maybe like if there's a mutation, this interaction is not happening now, so let me break that down and see what's going to happen. Yeah, we didn't do an experiment on this, but that's a great idea, yeah. Yeah, please.
I'm also curious in the first part that you use pairwise shortest path to like derive the features of graph. I wonder, have you tried other graph structures like cliques or like some, I don't know, it's like...
Yeah, we did actually, but they didn't work that great. Yeah, we did several things until we arrived there. Yeah.
So it looks like that when you're going through gene expression and protein expression, you're binarizing the features?
Uh-huh.
And then how sensitive is it to the binarization?
Yeah, we have that actually in the supplement of the paper as well. We did a suite of experiments on that. Like, what if we just use the continuous values as is? What if we use just the z value of the change? And what if we, at the end, what worked was like, if it is significantly different, use the z value, z score, but not, if not, then just 0. So it's like in between continuous and binarized. But we have the results for all these different setups. But it has an effect, yeah, certainly, yeah. Mm-hmm. Thank you.
Ruby, you have a question?
What, just a curiosity.
Mm-hmm.
Is there any way to include uncertainty in any of these methods?
Right now, no, but I think there could be, because if you bootstrap maybe certain things. Yeah, but for other things too. So you can get maybe estimate of like how uncertain the weights and so on, things like that. Yeah, yeah,
problem once you fix the
yeah, yeah. Yeah, but we haven't handled that. Thank you. These are very nice questions.
Any more questions? I know there are some causality experts in the audience. They'll probably catch you later.
Okay.
So
Maybe I'll catch them.